So, good afternoon, everybody. Welcome to this roundtable on the theme COVID and science. And first of all, let me thank this, let's say, incredible panel of scientists that was so kind to accept our invitation to participate in the roundtable. You already know them from the program, but let me briefly present them. So there will be Rui Moreira as president, Professor Rui Moreira, president of the European Federation for Medicinal Chemistry and Chemical Biology. Professor Gabriele Costantino, director of the Department of Food and Drugs of the University of Parma, and past president of the Medicinal Chemistry Division of the Italian Chemical Society, and actually vice president of the Italian Chemical Society. Dr. Emanuele Andreano, Monoclonal Antibody Discovery Lab, Fondazione Toscana Life Sciences. Dr. Neil London, Weizmann Institute of Science, Rehovot, and also member of the COVID Moonshot initiative, and we will discuss about this. Professor Armando Genazzani, University of Piemonte Orientale, member of the Technical and Scientific Committee of the Italian Medicine Agency, and also member of the Committee for Medicinal Products for Human Use of the European Medicine Agency. Giovanni Maga, Dr. Giovanni Maga, director of the Institute of Molecular Genetics and head of the DNA, Enzymology and Molecular Virology section of the National Research Council of Pavia, Italy. So, welcome to you, everybody, and it's really a pleasure, as I was saying. The idea of this panel and the idea of this discussion will be to consider the effects of COVID on science and the effect of science on COVID. In fact, during the last 16 months of pandemics, we have assisted to an unprecedented outcomes of results, and these months have been unprecedented for so many reasons and affected many aspects of our lives. As regarding research, they have been 16 months of incredible, outstanding, and unprecedented outcomes. But also we have witnessed a great impact COVID had on science, but also science reputation and science communication. So let's start on the probably most obvious question. I have to say before starting with the topics and with the question that each of the topics, each of the questions will be completely open to all of the panelists. So I will try to foster the discussion and stimulate it, probably addressing a specific question to a few of the panelists, but feel free to jump in and add your thoughts or consideration and your feelings in this regard. So let's start, I would say, with probably the most obvious topics. Vaccine. At this regard, where we are and what's next? What can be expected in the near future? I don't know if probably Giovanni or Armando would like to say something in this regard.
So please, Armando, if you want to start, let me know.
I will start. So the state of the art at present, I can talk about Europe, obviously, is we have 4 vaccines. 2 are adenovirus, obviously, and 2 are mRNA. Will more vaccines arrive? And do we need more vaccines against, let's say, the wild type variant? The answer is we possibly need more vaccines, especially for provisions. And it is likely that some of the same platforms will arrive, as well as more traditional protein-based vaccines will also arrive. What else will arrive? I think that vaccines which are more selective against variants will arrive in due course, and possibly cocktail of vaccines will also arrive. Most importantly, data will arrive. So we will know how long the coverage of a vaccine lasts. We still do not know this, so we're all guessing when we should be getting the 3rd shot. We don't know yet, and and really the extent to which the vaccine alone will manage to get us out of a pandemic. So this is what is here and what will arrive. I think this is quite important. Maybe there is the last thing which will arrive. This is very important. It's news of today, of yesterday, and maybe it's sort of not seen as important to most people, but a number of manufacturing sites are being implemented by the 4 vaccines which we have at present, which means that the availability of vaccines will be higher, there will be more competition, and they won't be limited. So it'll just be how many people we can vaccinate in a day, which will be the limit, but not how many vaccines we get. This is, I think, quite important. Maybe I can add something slightly more controversial, which is, do we still need adenovirus-based vaccines if we have the mRNA, which work well? In my opinion, we do. We do not know what the stage of a pandemic will be and what the incidence of coronavirus will be in September, October, whether it will come back. So the more vaccines and the more variety we have, the better to protect us from a new wave. Yeah. Giovanni, to you maybe.
Sure, no, I agree, completely agree with what you said. I just would like to add a couple of considerations. Well, the first one is about the future of the vaccines, because all of the vaccines that are now under development, either based on mRNA or DNA, for example, the one administered by electroporation, which seems to score fairly good, or even those composed of purified subunits, they are all based on a single antigen, the spike protein. However, there are now a number of studies showing that in infected people, you are eliciting antibodies against a wide range of immunodominant epitopes, which are also coming from other proteins and envelope several non-structural ones. And it seems that also those antibodies are, let's say, correlating with the ability of the patient to control the infection. So I think that the more that we will learn will also enable us to plan for vaccines including more than one antigen. For example, like those against influenza virus. And in this case, since some of these proteins, like the N protein, are extremely conserved, so they are less prone to mutate, we might even get to a point where vaccines will be really active against a wide range of eventual variants, because we know that the most variable regions are concentrated in the receptor binding domain of the spike protein. So I'm expecting really some exciting development in this field, the more we know about the natural immune response towards the infection.
Thank you. What do you think, any of you, what do you think about the intellectual property issue regarding vaccine and the point regarding the vaccination of all the world? I mean, in particular, developing countries, if we consider it's important for reaching a real level of coverage around the world. For instance, Gabriele, if you want to add something in this regard.
Yeah, this is a very crucial point, of course. As a matter of fact, at present, less than 20% of the world population has been exposed to at least one shot of vaccination, which is very little amount of the general population. And there is quite an excellent overlap between wealthy countries and our availability of vaccination. So I think it's so we cannot hope to reach the so-called and famous herd immunity if half or even more of the world population will be unvaccinated. So the point is to bring vaccines is a crucial one, but I don't think it will be I don't think it can be faced by addressing the point of intellectual property, because it's know-how the real obstacle to developing additional production in other countries, and not the patent protection.
And also the logistics, deployment and logistics of vaccination is much more complicated than, for instance, small molecule antiviral. So it's not only the IP and the know-how, it's also the cost of manufacturing and distribution.
Yeah, this is true. This is true. But if you consider that if you move to low-income countries, you always find a bottle of Coke or a cell phone. So bringing technologies there is not that difficult if you want to do it. Clearly, you cannot bring things that must be stored at minus 80 Celsius degree. But if you move to protein vaccines, as I don't know if Giovanni or Armando was saying before, I mean, this can be done. So I think the willingness to do is the best approach so far, rather than coping with intellectual property.
Yeah, this is exactly the point, because actually mRNA vaccine, mRNA-based vaccine, of course, seems to be based on a delivery system that is mainly due of liposome or something that require specific plants, specific reactor, specific device to be produced. I mean, in terms of flow and microfluidics, to obtain very particular liposome or delivery systems. That probably could be the hurdle in terms of technology. So to address the issue of delivering a vaccine that could be effective all over the world and even accessible to developing countries could be to obtain at least effective vaccine that could be also delivered with a simpler system. It could be done without requiring any particular technology in this regard.
If I may add, again, a very general consideration. So if we look at the case of poliomyelitis, even though we might, I'm sure, have maybe even in a short time a vaccine which could be delivered also under very problematic, let's say, social and demographic conditions, it will take time to get all the world population vaccinated. So I think we should not illude ourselves that this will be a goal that we can reach fastly. So we have to keep in mind that we need to cope with this virus for a few years at least, because it took more than 20 years to almost eradicate poliomyelitis, and Africa has been declared polio-free only last year. So it's really a matter of investment, money, logistics, as you said, willingness to do it, but still it will take really a lot of time, in my opinion. So that's not a problem that we are going to solve. I mean, the coverage of the world population in a very short time.
Yes. So Giovanni, just to you directly, a few months ago, I read a paper on Nature regarding the possibility to develop a rather general vaccine against coronaviruses. I mean, actually, this is a real pandemics, but we already have at least epidemics from SARS and from MERS, so other coronavirus. So what do you think? Is it possible in general, in your opinion, to try to think, to design and focus energy and research efforts in try to develop a rather general coronavirus vaccine?
The idea of a pan vaccine is not new. Also against influenza, there has been a lot of efforts to try to find a pan vaccine against all the influenza strain. The key point is to identify a very conserved neutralizing epitope, meaning some part or different parts of effective antigens that are quite conserved across all the different viruses. The idea of this broad spectrum coronavirus vaccine was exactly that one. So they were combining a very conserved portion of the spike proteins, which should be able to elicit antibodies active against many different coronaviruses. So in principle, I think it's possible. It could also be very useful in the long term, because we know coronaviruses are very common in the animal kingdom, and they have a very high potential of jumping species. So it could be that we might face in the near future another coronavirus, entering the human population. However, right now, I think the most important thing is to have really effective vaccines against this particular coronavirus, because it is now a pandemic virus. We know that pandemic viruses do not, fade away, but they will stay. So we need to find a way to change the perspective of these pandemics, in the sense that we don't have to think that we can eradicate this virus, but we have to make it manageable. We have to be sure that even though people can get infected, they will not suffer a severe illness. They will not be hospitalized or in very small numbers. So if we can reduce these epidemics at the same level of the influenza, seasonal influenza epidemics, which usually infect 5 million people in Italy, but in the 4 months kills directly 200 people and indirectly maybe 3,000-4,000, then it will become a manageable infection. And the only way to do that is to have effective vaccines and also effective drugs, therapeutics, but this we will address it probably later.
Yeah, thanks. In fact, this was the following point I was to discuss. We have, of course, and it's quite natural and understandable, discussed quite a lot of vaccine, because, well, we all need to be vaccinated to at least try to cope and try to control the situation. But also, beyond vaccine, besides vaccine, there are many other things that should be considered. And in particular, there is the role of, let's say, therapeutics. And at this regard, a few, let's say, important outcomes, a few important achievements have been done regarding a small molecule. There are a few of them in trial, for sure, and also many important results have been published. So, at this regard, I would like to know the opinion of NIR. The NIR in particular is involved into one of these projects, is the COVID Moonshot, but also Armando regarding the agencies and so on.
Yeah, so I agree that I think it's very important that in addition to vaccinations, we will have small molecule antivirals. One factor is what we discussed earlier. It would be much easier logistically to have fair and global access to small molecule antivirals, and it will be easier to get them to third world countries and to distribute them. And there can also be very good insurance policy for future pandemics. We're seeing now that inhibitors, for instance, of the protease of SARS-CoV-2 have virtually the same potencies against SARS-CoV-1. So had we developed a small molecule inhibitor and got it to past phase 1 for SARS-CoV-1, we would have something already ready to test for this pandemic. And in fact, one of the fastest developing therapeutic is a compound from Pfizer, which they originally started developing against SARS-CoV-1, and now they reignited that project, and they have already 2 compounds in clinical trial. One of them is IV only, but one of them more recently reported is also orally bioavailable. So I think this is very important. It can help mitigate the disease and stop hospitalization, especially we think an oral bioavailable anti-COVID drug is important since it can decrease the viral load before the patient needs to be hospitalized. There is remdesivir, of course, which is approved and is very effective, but since it's only administered via IV, the patient population that gets remdesivir for where the virus is the actual problem. Once patients are hospitalized, it's actually the increased immune response, which is the dangerous phenomena. So lowering viral levels is a little bit less effective in that stage. And so the COVID Moonshot that Gianluca mentioned is a sort of global collaboration which exactly tried to achieve that goal of a fair global access small molecule therapeutic against the main protease. And to do that, we try to crowdsource medicinal chemists around the world. So we started this project at the beginning of the pandemic. A lot of countries were under lockdown, and computational medicinal chemists were looking to help from their home. We were fortunate enough to have early access to the protease protein and to high-throughput crystallography at the Diamond Light Source under Frank von Delft and Martin Welch's leadership. Here at Weizmann, we set up an enzyme activity assay very quickly, and what we were missing were basically designs for what compounds to make and test. We collected and published to the public domain about 80 crystal structures of small molecules bound to the protease. These were all fragments, but they were good starting points to make new types of protease inhibitors. And so far, after about a year of this public effort, we have molecules which are nanomolar inhibitors of the protease with very good activity against live virus in cells. I think the important point here, besides the perhaps the implications for open science and an alternative pipeline for drug discovery, is that our compounds represent a new modality to inhibit the protease. So Pfizer's compound is basically a peptidomimetic. Their orally available compound is a more elaborate and more sophisticated peptidomimetic, but it's still a big and expensive molecule, which I'm not sure is scalable or will get eventually to third world countries. This fragment-based, structure-based approach led us to a very small compound, very low molecular weight, which is not similar to a peptide, is not covalent, as opposed to Pfizer's compound, and opposed to my natural inclinations. Our lab actually focused on covalent ligand discovery and design, but our lead series is not covalent. So I think it's a good example how such a crowdsource effort and open science can lead to new modalities and new therapeutics. And as a last point, one of the key features of this project was to develop. There's no financial incentives typically to develop drugs against pandemics or pandemic preparedness. Now, of course, after so many millions of people are infected, of course there are financial incentives, but for the next pandemic, it's not so obvious why a big pharmaceutical should develop the next generation drug. And so this open science, no IP project, we hope might serve as a template to enable academia or a nonprofit institution to develop the next generation of drugs without clear financial benefits.
Thanks a lot. So, Rui, it seems that collaboration is the key. So, in particular, what do you think? What could be done in Europe and in general within the European Federation to bring actors together and try to address such important challenges in, of course, in this situation, but also in the future?
Well, I mean, what we have seen in the past year and a half is really, it shows how collaboration is essential to get into very obvious answers in terms of therapeutics. And I mean, the case that the example that Nir just mentioned, the COVID Moonshot is just one of the examples. And I think if it wasn't collaboration and networking, probably the same results wouldn't have been attained in this very short period. So basically, collaboration is essential. I mean, there are additional areas of therapeutics that can be addressed, and eventually, for example, targeting, that's eventually an interesting point, targeting the host targets that are related to infection as well is very challenging, and probably any useful answer will come only from collaboration. So in terms of EFMC, so EFMC is strongly involved in promoting these collaborations and making it clear through our network of events. So that's something that we are obviously involved in, and we support any event that will make clear that collaboration is the basis for research in terms of COVID. So I think that's something that we could bring to discussion here is where else can we try to advance knowledge for targeting SARS-CoV-2? Is host targets are relevant or not? That's one point. The other one, which most of the public is aware of, is the question of repurposing drugs. So everyone heard about hydroxychloroquine over this year and a half. So it's a useful starting point to get drugs against SARS-CoV-2. So these are points that I would like to hear the panelists about.
I'd like to, in answer to that question, I'd like to note a paper that just came out in Science this week, I think, from Brian Schoiket's lab at UCSF. They discovered basically an artifact that is related both to hydroxychloroquine, but also many other drugs which induce phospholipidosis, which makes it seems in vitro as if it has antiviral activity, but actually you can develop it for in vivo administration. And this is a great example how very, a small investment in basic research could have saved billions in clinical trials that were wasted on drugs such as hydroxychloroquine. So I think, this research was also a result of collaboration between several groups in UCSF and a group from Novartis, if I remember correctly. And more basic science investigation like these could really save the public a lot of investment.
So if I may add also a very short consideration, because one of my interests is developing, let's say, antivirus targeting host factors. So relating to the question, are host factors relevant? Well, of course they are. And yes, it's challenging to find the right target. But if we consider that targeting host factors is the regular way to fight cancer cells, so we know that it is possible to find a therapeutic window of opportunity that can, have a beneficial effect on a diseased cell, meaning, for example, in this case, an infected cell, without harming, uninfected or healthy cells. And I think that if we want to escape, let's say, the always present problem of drug resistance that unavoidably will be present as long as we will start to use especially monotherapy against the virus, I think finding the right host target might help in, avoiding or at least reducing a lot the ability of the virus to escape from the kind of drug, because host cells are much less prone to mutate, especially if we target not protein-protein interaction between a viral and a host, but also some enzymatic activity in the host cell that is essential for viral replication. So I believe this might be, I would not say the future, but one of the most promising future approaches to develop even broad-spectrum antiviral agents.
Thanks a lot. There's a question from the audience from our very good friend Mario Varasi, that is also a very good friend of the ESMEC school. Mario said, no IP, no profit type of collaboration is great, but who's going to pay the development, in particular clinical development?
Yeah, I can try and answer that. So we thought of several models on how to bring this actually to market, not only as an academic exercise. And two obvious answers are philanthropy-based foundations, such as the Gates Foundation and the Wellcome Trust, are both interested in promoting such projects, as well as there's ACT-A, which is a consortium which involves both the Gates Foundation, the Wellcome Trust, the World Health Organization, and some other organization whose sole purpose is to sponsor, bring fair access drugs to market. So this is one direction. The other direction, perhaps you heard the U.S. just committed about $3.2 billion to promote antivirals, and bodies such as the NIH NCATS, the Unit for Translational Science in NIH, have the resources and capabilities of clinical development of such assets. So partnering up with national government-funded facilities is also a possibility to bring it to market. And then the last avenue, several big pharmaceutical companies are actually interested as not for profit, just for the benefit of humanity, are interested to help bring such compounds to market, if it's not against their own sort of pipeline. So companies that are currently not developing for-profit COVID-19 therapeutics are willing to talk about helping with clinical development. And the model there is that there will still be no profit except for what generic companies are making on making pills, right? So generic companies are still profitable without any IP. So a similar type of model where you go straight to generic can work in these cases.
Thanks a lot. So, Gabriele, since you are also a member of the relation between industry and academia, between also the Italian Chemical Society and industry, so what do you think? Is this feasible in general, well, let's say in Italy, but also in Europe?
I basically agree with what Near London just said, actually. I would add one thing in the context, specifically concerning COVID-19 or SARS-CoV-2. In the United States, as Near was saying, but also in Europe, also in Italy, and maybe Armando can elaborate a little bit more on this. Last month, I've seen really a lot of money coming from taxpayers used to promote clinical trials and clinical development, even of compounds that are patented and that are under IP protection. For example, we are paying as taxpayers here in Italy, we are paying, I don't know how many, but maybe Emanuele then can say something. Clinical trials for the use of monoclonal antibodies. We bought in advance more than 500 million euro in drugs without any clinical authorization yet. So this is a form of rewarding the intervention of companies in the field. So just thinking on IP is really a limiting view in the context of a pandemic. So I don't think this is the real problem. I think the problem is who is deciding where to put the money, because the money are here actually. So there are a lot of money inside, and there is a lot of know-how in the pharma companies. The point is how to match the two of things.
Yeah, indeed, I agree. I have to say that also a few private-public initiatives put a lot of money to develop drugs. Just to mention one of them, the IMI initiative bring quite a lot of money in this regard, but also one of the EU, one of the last Horizon call started a few projects. One of them is also involving a few Italian partners, just to mention the Escalate-V. So there are quite a lot of research, quite a lot of projects that have been funded. As you were saying, Gabriele, yes, the point is who is deciding, who is in charge of deciding where to put the money. That's the point. And Armando, what do you think as member of the Italian agency? This is, of course, this is a problem that need to involve you in the discussion.
No, thank you very much. Can I just go back a few questions ago? Because
sure.
I mean, it got so interesting that I thought I had something, an opinion on just about everything which was said. The first thing is that we said a lot of great things that can happen, and we can create a pan-coronavirus drug, we can create small molecules, we can change. The only thing we said we can't do is vaccinate the entire world. This is surprising, since we're talking to a very young audience. Yeah, we can change the future, we can raise the bar. Is it possible that the only thing that can't be done is vaccinate the others? We have managed to move from 100,000 people vaccinated to 500,000 in a week in Italy. Is it really true that we can't bring a €2 a shot adenovirus to Africa? I am not sure. I think if there is the will, we can do it. This is the first thing I want to say. I heard a lot of pessimism around this. I'd like to remind that variants and viruses move very fast. In a day, they can go around the world, and there are a lot of people dying in the underprivileged world. So I think we should make an effort to say we will vaccinate them, not we are pessimists that we won't be able to. I understand there are limitations, but there are hurdles, but I think we can do it. The second thing instead, I was fascinated about NIR's open science. And I have to say, this is a great lesson of the coronavirus science, in my opinion, which is in peace times, we can all be very personalized in our own labs, trying to make the most of our research and scooping the others and trying to publish on Science, Nature, JMedChem, wherever. In a war period in which we've got to be fast, if we work together, we'll be faster. Not only will we be faster, we will be successful. This is true for the project NIR said, but it's also true for all clinical trials we can think of, the Solidarity trial, the Recovery trial. All the trials that made a difference to us have been cooperative and have been very large. This is something that, as scientist myself, I find it hard to change the paradigm. No, I want to find a discovery and make it only mine. But those that have shared their knowledge fast and have cooperated with others have made a difference. Just think of corticosteroids, for example. Instead, regarding Mario's question and IP and clinical development and money, I'd like to remind everybody that most of the clinical trials which have given us information in this pandemic have been public-based. Most of the vaccines have been developed by companies with money which was given upfront by states. So it can be done. As everybody said, money is not an issue. It's the ideas which are an issue at present. This is how we can change it. Gabriele referred to the Italian Medicines Agency. We had a number of problems with the monoclonal antibodies. Maybe we'll talk about later, but we were unsure whether where they were developed was the right place, where clinical trials were informative or not informative. So we decided to finance our own clinical trials to decide which one was best, which was the best setting. The money was there. We are starting the trials. There is only one last comment I'd like to make, which is something I learned some time back when it was the Ebola virus, and also refers to Nir and also refers to Mario's question, which is finding an antiviral for a pandemic virus or for an epidemic virus has only one problem, which is that you need to be in a pandemic to be able to study it.
Yeah.
So it's not the money which is an issue. The problem will be finding the cases to study.
The patients, right? So
yeah,
so this is an interesting question.
But the question is also if people would have tried the first pandemic, maybe we'll have more compounds now ready to go directly into phase 2 clinical trials, right? I agree. Patient scarcity might be a problem, although at this pace, I'm sorry to say, it appears it won't be a problem with SARS-CoV-2. But even if happily, we beat this pandemic, there's no more patient to test the drug against, at least we have something on the shelf that we can immediately test in COVID-25.
Yeah, no, I agree. Preparedness, what to test is important. This is something we found out in March, April last year. So we had a lot of patients, a lot of ideas which weren't really backed up by data, and no real drug to test. So what Nir says is important. Maybe I'd like to say, although possibly the medicinal chemistry community might not be necessarily the right place to say it, but adaptive trials is also the word which we have learned. So creating one big trial in which drugs go in, once they either fail, and if they fail, you move them out and you take another drug in, and you can repeatedly test drugs one against the other. This is something which we found is very important, and Nir's platform and all the drugs that all these open research platforms are carrying forward are crucial for this strategy.
In fact, let's add something else to the discussion. Since we mentioned, let's talk about a bit more on about monoclonal antibodies. Emanuele, what's your opinion in this regard? What's going on? What's the, let's say, the next expectation in this regard in this field?
Well, I personally do think that there are a lot of the monoclonal antibody field has been exceptional in this pandemic, especially the quickness, the rapidity with which new monoclonal antibodies enter clinical phase from the discovery and development. This has been definitely unprecedented, the rapidity with which these antibodies were developed. And when in all the states where they have had more money to develop this kind of antibodies, basically they moved very, very quickly, and we have the case of Regeneron, Eli Lilly, Vir with GSK as well. I mean, they're so rapidly moving to clinical phases. It has been absolutely incredible. Now we as well, despite our limitation being a small institution compared to massive pharmaceutical companies, we managed to go from the identification of a human monoclonal antibodies to the development as a product within 3 to 4 months. And now that the antibodies has finished its phase 1 and enter the phase 2, 3, I mean, everything is ongoing. The major problem with these monoclonal antibodies obviously are the variants the VOC or variants of concern and variant of interesting that are rising constantly. Some of the monoclonal antibodies are still able to neutralize all the variants that are coming out, that are emerging. And for instance, there is the Regeneron, the Eli Lilly, some of those, they do not work anymore. There is the LYCOV 555 that actually have been revoked the emergency use authorization because it was not for futility in the clinical trial, so it was not working anymore because of the emergence of these variants. And this is probably the scariest thing for people that actually develop monoclonal antibodies. And is a constant race. The new variant appears, and the very first thing that you have to do is try to get this variant as a pseudoparticles as well as a live virus, and try to test your monoclonal antibody as quick as possible to be sure that actually the new mutation that appeared in nature will not impact your antibody. I mean, it's very exciting in terms of field, very challenging. But I think they are showing to be incredible valid molecules, especially at the early stage of the infection. Now, I think just a week ago, there have been some data from the Regeneron cocktail that actually seems to be also effective in reducing the mortality at 28 days in seronegative patients, for instance. And this is also, I think, in hospitalized seronegative patients. So this thing I think is a very, very good news. But everything is moving very fast, and we really need to have a lot of bullets in our arsenal to try to cover all the variants that are coming out.
Yeah, that's true. That's definitely true, correct. But well, in general, I agree. But the point is, if you need, since you discuss about the Regeneron case, if you need to subministrate, let's say, grams of monoclonal antibodies just to obtain a very limited efficacy, a very limited reduction, and also in a setting that is that needs to hospitalize the patient, in comparison with let's say limited benefits that you can get. So do you think that it's still worthy?
I mean, I think it really does depend on the case. The Regeneron cocktail, it really does depend on the potency of neutralization of your antibodies, which translate into the amount of antibody that you need to administer to the patient in order to have and to observe some efficacy. Regeneron goes with different grams, for instance. In our case, we tested side by side our antibody with other competitor antibodies, and we've seen that our antibody is working very nicely in neutralization in vitro. And we are starting to give the antibody intramuscularly, very low dosage, 100 and 400. Actually, the phase 1 had 48, 100, and 400 milligrams, just one injection. And the pharmacokinetic of the antibodies was so nice. After, like, let's say less than 48 hours, you already had a nice peak in the serum of these patients, and after 8 days, basically, you had neutralization titers that are far above what vaccination induced. I mean, in our case.
Sorry, can you repeat? I didn't hear it. You get the peak after?
So after 48 hours, we already had seen a very nice peak in terms of PK, and at 8 days we reached the max peak, and we performed neutralization assay with those sera, and the neutralization titers were far above those induced by vaccination, for instance. And I think with one injection that can be given intramuscularly, i.e. can be given at home, can be given at your doctor. It doesn't need to be IV, so you don't need to be at an hospital setting to do so. I think this is definitely worth trying to get these antibodies out there with this scenario. Obviously, if you have to feel healthy people, or let's say people at the early stage. You have to fill the hospital with people at the early infection of COVID-19 because they need 2-3 hours intravenous injection. Obviously, that gets the thing very difficult. You need nurse there. You need a lot of time and space that sometimes is not possible to have because hospitals are so clogged. I mean, it really does depend on the molecule and on the way you're going to do this. The way we're trying to do this should be 100 milligrams, so feasible for low middle income countries as well as high income countries. And by giving it just IM, so intramuscularly, you should bypass all those obstacles that are introduced with intravenous injection. So to answer you, yeah, I think it's still worth it to investigate this molecule.
No, yeah, yes, definitely investigating. It's still really worthy, and of course, once you have very effective and potent molecules, of course, they are a solution. I guess that Armando would say something in this regard. Yes.
No, I agree with what has been said. I think antibodies are a good armamentarium in a non-pandemic setting. So when you have a few patients, then antibodies, especially the IV antibodies, are great to reduce the amount of people which might somehow go to ICU or die. I also think it gives us a great lesson in drug discovery. So we should think of the disease first at times and understand exactly where we want, what sort of phase of the disease we want to tackle before developing a drug. So in this case, the first tests for the antibodies have all been in hospitalized patients, which was the wrong place to do it. Then they moved back and they said, okay, it will be home-based patients, and there was a minimum benefit, as you mentioned. And now, finally, Regeneron comes with data showing that there are some patients which go to hospital, so are severe, have let's say a high chance of progression and are seronegative, and there we are finding a quite high benefit. So developing a project from knowing the disease, which obviously was not possible in a phase in which we were learning alongside, is a great lesson that I have learned for drug discovery.
Yeah, Gianluca, if I can add just a little bit.
Definitely, yes.
I think this is one of the most relevant points in the antibody discovery. So the question of seronegativity of patients. So I think this was one of the less under considered observation that Regeneron did already in January, December, when they went out with the paper. So the answer is only for people that are naive against natural antibody for SARS-CoV-2. And probably this is the reason why. So I'm not sure that the point is that antibodies should be used in a non-hospitalized setting. I think they can be very effective in hospitalized and severe patients, provided they have not mounted an antibody response by themselves. This is the point. On the opposite, if they are seropositive, so they are already competent, administration is useless, simply useless. It's like giving a bottle of water inside the sea and see what happens, just because you have already your immunoglobulins in the circulation. So I think in order to exploit the great potential of this treatment, it's crucial, to my understanding, stratify the patients in the right way. So not all of us will be appropriate for the treatment. And saying for patients at risk means nothing, actually, if you don't have biomarkers that define the risk for a given patient. And to my understanding, is the seronegativity that define the risk for a given patient. So I would like to hear the comments from Emanuele, for example, or the others concerning this.
If I may, I do have a couple of comments. So definitely, we have to think that these patients hospitalized with COVID-19, they receive depends obviously on the state and everything, but they might receive a huge amount of corticosteroids, and this will definitely dampen the immune response, and sometimes they might not elicit an important antibody response capable to clear the viral infection. So that's why in those patients probably it's critical to administer passively antibodies so that they can kind of cover the whole thing. Sometimes monoclonal antibodies, or actually antibodies elicited following infection, can also enhance the disease. There are different scenarios like the antibody-dependent enhancement of disease, for instance, or they can elicit autoantibodies, for instance, or they can induce cytokine storms and scenarios like this. So giving 8 grams of antibodies may be worthy, but really, I think, does depend on the clinical scenario. We thought about this a lot, for instance, also based on previous experience, like with the SARS-CoV, the first one, where the ADE or antibody-dependent enhancement of disease was observed or postulated. It was postulated here as well. So we knocked off the FC functions, for instance. So our antibodies not elicit any kind of production of pro-inflammatory cytokines, induction of ADCC, or any sort of activation of the immune cells. And I think this could be also relevant for this audience, especially engineering of the FC portion of a molecule of an antibody can be critical for the effectiveness of the molecule itself. So, and also, if, for instance, when the patients are extremely severe, then if they have extended damage to tissue or the respiratory tract, in that case, obviously, a monoclonal antibody will not restore that kind of situation. So I think it really does depend on the clinical scenario that you face, whether or not a monoclonal antibody could be a benefit for the patients or not.
Yeah, definitely. I don't know if Armando or Giovanni or Nir would add, or Rui would add something in this regard.
I have just a basic question, actually, because I'm not an expert in antibodies. So, but thinking about what have been done with HIV. So I'm wondering whether we are not already at the point that we can also program our monoclonal antibodies in order to overcome the problems of the variants just by mapping the neutralizing epitopes that patients are developing, and then within them identify those conserved portion of the spike protein or whatever other protein that we find to be eliciting neutralizing antibodies, and then design monoclonal antibodies that can eventually then be active against those variants that we know are almost concentrated on specific parts of the protein. So I don't know whether Emanuele can maybe comment if we are already at the point to do this.
I mean, HIV, the GP, the glycoprotein, it's quite different compared to the spike protein. GP 120 has a huge glycan shield. It has very thin spots where antibodies can go there, or the patient, after several years of infection, develops antibodies with very long CDR3 that allows to go around the glycan shield and still be able to neutralize the virus. But sometimes it is because of the extended CDR3, they get to be also autoreactive and stuff like that. So with the SARS-CoV-2, the spike protein also is heavily glycosylated, but at the same time, there are spots like the NTD and the RBD, obviously the two most important ones, that allows the binding. It has been very deep investigation of the different epitopes and spots. There are now defined different class of monoclonal antibodies, different nomenclature. There's no one single nomenclature. For instance, Pamela Bjorkman lab, they kind of stated they are called like class 1, 2, 3, 4. depend if this is the RBD. So they do like 1, 2, 3, and 4. There is Ian Wilson, Dennis Barton lab, basically at the Scripps Institute. They define that like A, B, C, D. So there's been a lot of defining of possible epitopes that induce neutralization. The problem is that the polyclonal response following infection and possibly following vaccination as well is strongly directed towards certain epitope. And because of this high pressure on the RBD of the virus is evolving, obviously, has evolved to evade this immune response. And some of the very initial molecules, like the LY-CoV016, is one of those very predominant antibody elicited and has been quickly knocked off. So I think there's a lot going on there in the antibody discovery field, and the HIV scenario is super interesting, but possibly a little bit different. So yeah.
Okay.
But
Thank you.
there is a lot of work that has been done trying to define neutralizing epitopes.
Okay, thank you. So, Rui, well, it seems that, it's not just now, even since many years ago, there is a world of therapeutics that seems to be aimed in biological therapeutics. There is still space for room for medicinal chemistry, and which is the role of the medicinal chemistry in this scenario? I think your microphone is muted.
That's like walking on very thin ice. So in this particular case of COVID-19, so SARS-CoV-2 infection, if you go back to the beginning where all the research started, both vaccines and medicinal chemistry were working side by side, and suddenly vaccines took the lead, and basically this is now something that is biologically oriented in terms of preventing the infections and treating the infection. But there is room for medicinal chemistry because we still need tools to overcome the resistance and the new variants. So there are areas where medicinal chemistry can bring new tools. One of them I've mentioned at the beginning. That's something like interfering with the host targets that can be related to infection, but also modulating the host immune system. These are more challenging areas, but they can lead to new chemical tools that can be useful to treat COVID. The thing here is that the results will come later, and translation from basic research to clinical trials will take longer, I think. And that's something that is typical with small molecules. I mean, even as Nir mentioned, I mean, compounds can be developed with a strong focus on oral bioavailability, but there is a long way between the discovery and the developing and translation into clinical trials with small molecules. And so there is a room for medicinal chemistry, yes. The results might come later than compared with vaccines, but they will be very important in terms of addressing questions like, or issues like variants. So that's something that I think we should consider as a strong possibility.
Thanks a lot. Of course, my question was thought-provoking, and I definitely agree with you in this regard. And also, Gabriele, probably you would like to add something to this topic and to comment something more.
It can only be a bit provocative here, in the sense that, well, of course there is a room for medicinal chemists, no doubt. But we should also consider that this is an acute and auto-limiting infection. So clinically, as far as I know, or we understood so far, this is an infection that took, I don't know, 1 week, 10 days, and then disappears. So person either recover or go to exitus, unfortunately. No? But it's a auto-limiting infection. And when you have an auto-limiting infection, it's extremely difficult to assess the efficacy of a drug in a phase 2, phase 3 study. That's the point. Because you cannot take the natural history of the disease as a surrogate for control group. You have just to take people and possibly design clinical trials, randomized clinical trials, and so on and so on. But with a percentage of people that develop a serious disease, which is very low. This is the reason why monoclonal antibodies so far didn't get a significant impact in the disease, or the same as remdesivir, for example. So you cannot decide. You don't have a real clinical outcome to decide whether your approach is effective or not. And I think this is the main limitation for a small molecule approach in the next few years. It's not an issue of targets or so on. So I think targets will be found either host-based or viral-based. They will be characterized and so on and so on. The point is to find a good clinical setting to assess the efficacy of the treatment. That would be the major obstacle to the development of medicine from companies, to my perception.
Yeah, I definitely agree. And if I can add my 2 cents to this point of discussion, actually, besides small molecules that of course are still important, and in my opinion will be ever important, because there will be a few scenarios where we would be not able to develop biological therapeutics. But there are, in my opinion, 2 other aspects that should be considered and could be open to the role of medicinal chemists. The first one regard the so-called conjugate. So the conjugation of antibodies and the payloads, it's something that is gaining a great importance in a few fields, in particular in the cancer therapeutics, and could be important even for other fields of research in the future, including viral infections, and also in the developing of let's say of formulation, of proper formulation, the medicinal chemist could be very important. But there is another important aspect, and this is related to the recent changing of the name of the EFMC into European Federation of Medicinal Chemistry and Chemical Biology. It's the role that small molecules have in discovering, studying, and investigating new targets, including, of course, targets that are important for instance, the host recognition and the mechanism of infection of viral agents. What do you think, Rui?
Well, SARS-CoV-2 is exactly one of the areas where the continuum between medicinal chemistry and chemical biology is most clear. So we are at the early stage yet, where we are developing chemical tools to understand how virus work, how it interacts with the host, and that's, I mean, basically we are more close to the early discovery chemical biology area. I think that's why we are very far, well, in terms of having a very long pipeline of compounds. We still to understand how the virus works and how it interacts with the host. And of course, in terms of developing and understanding the molecular mechanisms, that's where chemical biology can bring a very extensive knowledge. And because the other point that I think it is important, compared with other infectious diseases, we still lack a lot of that data. So data is still building, and data will build from the basic side as well from the clinical trials. And I think that in that regard, the things like developing very robust phenotypic assays for screening against SARS-CoV-2 and developing compounds that are really good tools to engage with the targets. It's going to take time, so it's an exciting area. I don't know how much time it will take to enlarge a pipeline of drugs, but that's something that eventually other ones can comment on that as well.
Yeah, definitely, I agree with you. And of course, there are a few new emerging topics and new trends in medicinal chemistry. For instance, today in the school, in the morning session, we had the PROTAC session. Well, the protein targeted could be an approach even in antiviral. Of course, a few of the protein could be directed with this approach, and there could be the idea and a proper small molecule design and directed to degrade something, some protein that could be important for therapy in general. So, but since we mentioned the variants, so let's talk about mutation and variants, Giovanni.
Well, I mean, forgive me if I'm stating the obvious, but as you all know, genetic variability is an intrinsic natural feature of all viruses, particularly RNA viruses. So SARS-CoV-2 is mutating, for example, less than the influenza virus, much less than HIV. But of course, given the fact that it's circulating all over the world and there is a very intense, generation after generation of viral population, the virus is accumulating. I would say right now we know about 100,000 mutations overall in the genome of SARS-CoV-2 and scattered among hundreds of variants. For example, in the UK, the public health system is monitoring, I think, 14 variants divided between variants of concern and variants under investigation. Just to say that variants are the rule. Now, the point is that under the selective pressure to become, let's say, more successful, and for a virus to be more successful means basically to be able to spread faster, so to be more contagious, not necessarily more severe, or let's say giving a more severe phenotype, but just having a better chance of infecting hosts. Then we have seen, and we will see also in future, the emergence of a few variants of concern. We all know them, and particularly there was the, let's say, Kent variant, the one that has been isolated in UK, that became dominant. Even at the very beginning of the pandemics, there were the 614 variant that, since was more infectious than the original virus, was spreading very quickly all over the world. Now we have these Kappa and Delta variants, isolated the first time in India. And all these variants, they have basically some common features. So they accumulate amino acidic changes in some particular hotspots at the level of the spike protein, basically mostly anti-D and receptor binding domain. The consequence of this is that these variants are more contagious, meaning that if they come in contact with our cells, they will have a better chance of infecting the cell. That doesn't mean that if we still use the, let's say, preventive measures or face mask and distancing, social distancing, those virus will be more dangerous. No, but the point is probably there is the potential to start an infection cycles with a lower viral load, and as a consequence, since these viruses are replicating more in the body, probably those people that are who are infected will then release higher viral loads, so they will have, let's say, more potential to infect other people. Now, what is the point at which we are standing now? We know that the Delta variant is becoming dominant in UK, is spreading quite rapidly in other countries. It is more infectious, more contagious than the previous variants, but there is no clear indication that it might give a higher risk of hospitalization. I mean, there is, but especially for naive people, not immunized people, because as I said, since it is more contagious, it can replicate more, and then you can have higher viral loads in your body. We know that a single vaccine dose is still giving some protection, but this variant is more capable of breakthrough infection in people who are vaccinated with a single dose, while the full vaccination, and this has been demonstrated both from epidemiological studies and from laboratory studies, is still very effective in preventing severe disease. So the point I want to make is we will always see probably the appearance of variants. The impression is that selective pressure that is now, selecting these variants is always focused on the spike protein, even though these viruses have, many other mutations in many other genes whose functions are still not very clear. But if we can achieve a good immune protection through vaccination, and hopefully very soon also having some therapeutics effective against these viruses, then it will not be so important the number of new infections. It will be, of course, but if the number of new infections will not correlate anymore with an increase in hospitalization or in deaths, then, as I said before, we might get to a point where whatever the scenario will be, it will be manageable. So we will not be again into an emergency situation with lockdowns and so on. However, right now we are still quite far from the point, because we need to have a almost full coverage of fully vaccinated people. So these variants are really of concern, because they can spread among population that is not vaccinated, for example, youngsters that are still missing, a lot vaccine coverage, and continuous circulation, especially in people that probably will be asymptomatic or showing very little symptoms, will probably foster the selection of additional variants. So the priority, in my opinion, is that we have to be very careful in trying to limiting the diffusion of these variants by sequencing, by monitoring, by making, a stronger surveillance, and we have to complete the vaccination as soon as possible. Then we probably can change the scenario, and I don't want to say we will not be so worried anymore about new variants, because we never know what kind of virus can, pop up, but surely we will have a change in paradigm. We will not be counting anymore total amounts of infected people, but we will be counting total amount of people having severe disease.
Gianluca,
Thank you. Sure.
A question to Giovanni, because what he was saying is very intriguing to me. The first question is, okay, we have to keep vaccination, and no doubt. What about vaccinating pets? animals? Because this is another reservoir, no?
This was actually the question I was to ask.
And sorry, and the second one, so you can as you say, as you said, the virus is accumulating a mutation in the RBD domain. Does it mean that somehow or sometimes it will bind to another carboxypeptidase instead of ACE2?
Okay, so for the first question, I mean, so far there are no real documented transmission from, domestic animals, with the exception of minks. We know that this coronavirus can infect feline or dogs, but it doesn't seem that the virus can go back to humans. So right now, unless we will see, really a potential for these animals to transmit the virus to humans, back to humans, of course, we can think about also vaccinating pets, but it doesn't seem to be a major route of infection. I mean, the major route of infection is human population, which is not vaccinated. So I would say, to the best of my knowledge, this should not be a priority, even though the fact that this virus can infect animals probably means that we will not be able to eradicate it, because it's not a virus with, let's say, unique human circulation, at least in this phase. For the second question, it's very hard to say. I mean, it depends. If random mutation, a virus strain will be able to change the receptor usage and having an advantage, because the point is, will the change of the receptor make this virus more successful in terms of spreading, and so having more generation at the other strains? Theoretically, is possible. I'm not aware of many examples in nature, because it doesn't necessarily mean that binding to another protein rather than ACE2 might be of great advantage to the virus. It seems that the pressure for this mutation in RBD is more driven either by the immune response or by selecting higher affinity for the receptor, which is not, again, a general rule, because, for example, the Delta variant doesn't seem to have a higher affinity for the ACE2. So it's still not clear why it is more contagious. But, I cannot say yes or no. I think it's probably not very likely. But it also will depend on how long the viral will circulate among a huge number of people, because then we'll create, the right condition for generating more variants.
Yes, also, just to add something in this regard, another part of the problem regarding what Gabriele was asking is not just considering directly the effect for humans, I mean, in direct term, but also the global impact that it could have on society. I mean, if we had to, let's say, kill large number of animals, considering, for instance, the case that happened in, I cannot remember, it was Denmark, perhaps, or another country, or if a lot of pets are being interested by the pandemics, of course, this could be a very important problem, I mean, also for society. So this is another part of the problem, considering the One Health concept. That's why also it could be considered and important. Probably in the future, having something that could be used not just for humans, but having something also for animals, in particular for pets, and in terms of therapeutics or vaccination could be important, not just for the spillover problem, but also for the problem itself, that having pets and for people who love pets who care for pets could be important for.
You know, there are already, I think, a couple of vaccines against animal coronaviruses. Not this one, but coronaviruses are spreading, you know. So, yes, I mean, I agree. But let's say, think about the flu, the avian flu problem. So when we had, you know, the outbreaks of avian flu with potential to spread into humans, we had to cull tens of millions of you know, poultries with a very huge economical loss. So for example, in Italy in 2007, I think there was a very huge outbreak of avian flu virus, and that was destroying really millions of poultries. And so the point is always, the one health problem is always played at the human-animal interface. I think we have to first see where the danger lies for humans, and then, of course, we can, you know, think about the wellness of our pets. No doubt about it. But in this particular context, I think priority would be to try to prevent as much as possible not only this pandemic, but also future spillover events. So if vaccination of animals, whatever it could be, is important to protect human beings, and this is my opinion, that's sure a priority. And then, you know, following all the other steps.
Sure. Thanks a lot. So let's move to another important point. At the very beginning of this discussion, I started saying that the last 60 months of pandemic have been unprecedented. But what is unprecedented is not just the speed and focus with which the community responded to the pandemic, but also the singular willingness of scientists all over the world to share new ideas and data immediately and transparently. And this is, of course, it was very good, but also gave rise to what we have learned to call infodemics. So there was a lot of information. A few of them were right. Many of them were fake news or misleading news. So what do you think? And this is directed to all of you. What do you think could be the lesson we should have learned and actually learned from the pandemics as scientists?
Well, I mean, I can.
Rui.
I think there are different levels of that problem of the infodemics. So if you think about the scientists, there was really nothing wrong with the way scientists communicate the science, even with the larger audiences. I think that the way the problem came up with how that information was perceived by larger audiences and politicians as well, and I think that was even for the media, and that's the lesson that we need to take is that probably in terms of media, I think we need to, or the media need to learn how to understand the information that scientists convey, and so basically the question of fake news is something that is in a way out of the circle of scientists, and I think we should not mix things together, and because it's something that is very difficult to fight against. And basically, my idea is that nothing wrong went with the communication made by scientists. I think the way the information came to the public was correct. The perception from the public might have been not the best one, and of course the one from politicians as well. how to solve this in the future, I'm not sure.
Gabriele.
Yeah, of course, we don't have answers or solutions how to solve the problem. My personal concern is more regarding irrelevant publication more than fake news. So there has been really a lot of papers out talking about whatever, putting the keyword COVID, SARS, and so on and so on and so on. And this, okay, now we are able, at the moment, we are able to discern probably the good from the bad ones. But I imagine what happens in 10 years time when the future students, the future PhD will go to the database looking for what has been published in 2020 or 2021. So there is really a lot of papers that whose significance and relevance is really difficult to. And this is probably also a responsibility of the editors. So I do understand very well that there is the need to publish, the need to share, also the willingness to appear. But this is a responsibility of all of us. I mean, so there is really an infodemy. Apart from fake news, this is a different thing, okay? But probably we can cope with fake news more robustly. The irrelevant, the unnecessary, the sloppy publications are the real risk, to my perception, in this context.
Yeah, can I add something?
Sure.
I agree with everything that Gabriele said. So a lot of things which should not have been published have been published, and maybe too many things. But I think really that we lost an opportunity, and I think we still have a chance to go back to that. So science and medicine are uncertain. So opinions are certain. So if I see the way that scientists have communicated, at least on Italian TV or on international press, we have tried to communicate, and the public wanted certainties. So they wanted us to say hydroxychloroquine works, hydroxychloroquine doesn't work. Vaccines do only good, vaccines do only bad. The truth is that science is uncertain. There is a famous saying that says 50% of the things we believe are true now will not be true in 5 years' time. This was a famous editor of the British Journal of Medicine, let's say, of the BMJ. So we failed somehow to say that we had doubts in what we were saying. And so we had, for example, experts on one TV channel saying one thing, and experts on a different TV channel stating a completely different opinion. And neither of the two said, well, I am uncertain of what I say. They all tried to flag that what they were saying had to be right. Because it's true, the public is obviously lay public. They do not read scientific journals. They do not know why those opinions have developed in that manner. So they just want an easy message. And this is what a lot of scientists that talk to the public try to give, an easy message. And I'm afraid science is not easy. And I'm afraid there is nothing is straightforward. And I think now it's the time to tell the public that actually when we have in a pandemic, we are building knowledge. All the knowledge today says that this is right, but tomorrow it could be different. So other things are opinions. I feel strongly about this because I have seen a lot of people that try to sell certainties for things I knew were completely uncertain and were their own personal opinions. And I've seen a lot of damage done. No, I'd just like to, again, because we have a young audience. So having had 2 or 3 experts that said that azithromycin, an important antibiotic, would have cured coronavirus, okay, will leave a huge burden of antibiotic resistance on this planet. And that was an opinion. That was not a fact. And the people that sold it as a certainty. So this is what I feel. Sometimes it's true that publicity has to give easy messages, but science is not always easy.
Yeah, I agree, and of course there are a lot of layers of discussion in this regard, because, well, first of all, in my opinion, an important role and responsibility should be ascribed to policymakers from one side, but also to, let's say, general newspaper, because a lot of general articles are being based just on archive, bioarchive, medarchive, chemarchive, so preprint server journals, so without any reviewing process, without any discussion on this regard, just to make sensation, just to make audience, just to obtain, let's say, a wide audience in this regard. And yes, of course, the democratization of knowledge is an important possibility, but also, well, we are now in a time when anyone is convinced that doing a quick Google search of about 5 to 10 minutes is could be comparable to a master degree in biology or medicine or whatever, or chemistry or anything else. So this is an important problem. On the other side, in my opinion, the limit is the fact that we have realized that scientific method is not being taught anymore in school. So people is not aware about science. So Armando, you said that science is something that is based on hypotheses, on data, and of course, on confutation of hypotheses when data doesn't support them, and people should be better taught regarding the scientific method and know better in this regard. We had, as scientists, also our responsibility, because for many years we have been closed into ivory towers, and now we need to be much more clear in this regard, but avoiding, in my opinion, the easy publicity and easy, let's say, rewarding that could be obtained from the wider audience. Giovanni, probably, I was saying that you were nodding and you were probably would add something in this regard.
Yes, well, because of course I feel partly guilty since I've been involved, sometimes even not willing to do, in the dissemination and information of the public.
You were always very, very precise and moderate, so well, I wasn't referring to you.
No, but I mean, so I agree with everything that all you and the colleagues said. I would like to add just a couple of considerations. The first one is that we have a system of media communication which really sometimes makes it very difficult to be, as Armando said, very clear about the limits of science, because the system works like that. You get a phone call and they say, well, 5 minutes ago I spotted this paper on a medRxiv, and please give me a comment, or in 10 minutes we will have you on the screen if you want to make a statement about this decision from the government, whatever. So very often you don't even have the time to be aware of what they want you to comment on, and these are those instances where I usually refuse. But other colleagues, I must say, do not, use the same caution. And what we see is that in some instances, even the I would say the fascinating word of the presence of the media is in some way making those colleagues overstating things. I mean, I completely agree that sometimes you hear people saying this and that as they were, proven facts, while they are just hypotheses. And this problem is emphasized by the way the media world works, because they want to have, first of all, yes or no answers, and also they like to have debate. They like to have people with opposing let's say vision, because it makes audience. I mean, I remember personally I've been asked by a major journal to be interviewed, and they told me what I had to say. So we contact you because you must be the one that is saying this. And I said no, because I mean my opinion is different. But this is the let's say the environment we have to deal with, and this is a disaster, of course. So it's true the scientists must how to say talk to people, but not all scientists are also communicators, because communication is a specialty. I mean, there are professionals who are entitled to do real communication, and the problem is sometimes media do not use these professionals. So in my opinion, communication by scientists should be done in the right places at the right time, meaning also in schools, in events like this one, just to try to educate people. And in terms of public communication, we should really stick to what we know and always say, this can be, but it's not sure that it will be like that. And in my opinion, the real problem is that within this plethora of voices that the media are, collecting, because every talk show, every news must have the expert, maybe 10 times a day, 10 different experts. So they call everyone, even people that are perfectly, let's say, extremely good professional in their own field, but might not be as much as expert in those topics that they are interrogated about. What was missing was the official voice. In many other countries there was then the synthesis point, the institutional voice that says, okay, we know this and that, we don't know this and that, but at this point what we know is this and this, and we have to do this and this. And that kind of voice, in my opinion, has been missed. So I think part of the problem is that also institutions have lost a big occasion to be able to engage people in the right way. I mean, just as a last consideration, the WHO, after the Ebola epidemics, the last one, set 10 basic rules for public engagement, because it was clear that if we want to stop an epidemic, we need the collaboration of the population, of course. And one of the pillars of these rules is communication must be made by someone who has authority, credibility, and so that can be trusted by people. Obviously, 100 different voices cannot satisfy these rules. So if I can say something, we really need to have not a single, but a very authoritative voice that then give the final answer to the best of our knowledge to the public, because otherwise we will never get out of this let's say devilish loop of people saying whatever they think in the morning and in the afternoon. Sorry if I was a little bit harsh, but this is what I'm feeling.
But I definitely agree. In particular, we experience a few very important cases of this kind of bad communication, let's say, of inefficient communication. Just to mention one of these is the communication regarding the AstraZeneca vaccine, of course. We all witness a very, let's say, strange communication in this regard. No, Armando, what do you think? Did anything go wrong with the communication with the AstraZeneca vaccine?
I think everything went wrong with AstraZeneca.
In fact, was provocative.
So, and this is maybe what I was saying also, taking my responsibilities. The AstraZeneca vaccine arrived with a study on 7,000 people, and they were more or less all under 55 years old. So the first thing that we did in Italy was, and in Germany and in France, was to say initially we would use this vaccine under 55. And so the communication went out that it was dangerous for the elderly, not that there was data only for the young. Okay. So at a certain point, we had data also in the elderly. So we changed it. And the communication went out that we changed our mind and we were wrong initially. But we were not wrong. We used the data we had. And just for, I mean, as a laugh, although it's not very, very funny at all, suddenly we become aware of pharmacovigilance, that the risk in young adults is higher than in the elderly, and obviously the risk of COVID is lower. So we say that the vaccine should be used just in the elderly. And this is data-driven, is what I was saying. We don't change our mind. Data changes our definitions. But again, it went out wrong that the AstraZeneca vaccine was dangerous and that we did not want to put at risk the young, but we could put the elderly. So it is difficult. And Giovanni said something which I think is right. So sometimes communication has to be done by professionals and by credible people. If we all communicate, if we are not professionals and we are unable to put the right word in the right place, there is a lot of damage that can be done. This is true. And yes, and obviously media can't do anything but look for divergences to sell newspapers or to have audience. And we should be aware of this. But sometimes things happen.
Yeah. Sure. Well, just to mention a joke. Yesterday, I heard a joke in a local radio that's saying that Pfizer was developing a vaccine against the AstraZeneca vaccine. So it could be next frontier in this regard. So just to mention another point of, let's say, bad communication that also involve policymakers sometimes regarding the so-called Sputnik vaccine. So in my opinion, of course, does it work? Probably yes. Do we have enough data to say that it works? I don't think so. So what do you think, Armando? The European agency and the Italian agency being involved in this discussion, and also Giovanni, Emanuele, Rui, and Gabriele, please add whatever.
Yeah, no, I'd rather not speak a lot about the Sputnik vaccine. That was also bad communication. Whether it was malignant or not, I don't know. There have been rumors that they approached the EMA and had submitted the dossier. They hadn't yet, so forth. The evaluation of that vaccine is ongoing. Probably it works, as you say. We have some rules and legislation. They have to do things in a certain way, following particular guidelines. We have we accept things from some districts, regional districts, and we have to instead inspect other districts because it's the rule. This would apply also to the US. That was also bad communication because it's sort of it gave it for granted that a Russian vaccine would have been different from all other vaccines. Instead, it was treated just the same, and it is being treated just the same. I'd just like to remind that the Sputnik vaccine is an adenovirus vaccine, like the AstraZeneca and the Johnson & Johnson, and therefore, it will create that particular cluster. It won't add that much to the diversity of vaccines we have.
Thanks a lot. Giovanni, do you like to add something, this one?
No, I think there is no, there's no real news to report. As it has been said, this is a vaccine that is being evaluated. The initial publication indicated that it might be effective, but of course, probably we need a little bit more data about different things, production, also not only efficacy. It is adenoviral vectors, which is also particular because it uses two different adenoviruses for the two doses. So let's say it's a sort of not heterologous vaccination, but in principle it might be, because you have one vector for the first dose and a different vector for the second one. We will see. I mean, let's see. Apparently, we are not aware of its efficacy against the variants, as far as I know. But my point is, the more vaccine we have, the better we are, equipped, especially because there are countries which rely on this particular vaccine for their vaccination program. So if we can demonstrate that it works, I think it will be a very good news. I mean, we have seen also for the Chinese vaccine that in the field they failed, at least the first generation. So probably those that are being developed now will be better. We don't know, but if we want to vaccinate the world, we need to have more manufacturers, because 2 or 3 big pharma companies will never be able to cover all the needs.
Sure, and this will open actually a discussion in which one of the panelists should be, let's say, Farmindustria or whatever, but it's a matter for another event, and in particular regarding the Italian situation. So we moved to the end of this the discussion. So I would like to ask the audience if there are any questions for the panelists. Otherwise, if anyone else has anything to add, any other point to add to the discussion, I don't see any question from the audience. So let me thank you again for your willingness, for your availability, and thanks a lot for this very stimulating and interesting discussion. I really enjoyed it. I appreciate it. I'm pretty sure that also the audience, as you know, we were live, and this event was broadcasted not just on the platform, on the website of the ESMEC school, but also was broadcasted using the Facebook page of the Medicinal Chemistry Division of Italian Chemical Society. So sometimes we take use of social media to do the right thing. So thank you again. I really hope that you will have time and chance to follow the rest of this edition of the school, and I'm pretty sure that there will be soon other chances to meet in person to other events. Thanks a lot, and thanks to everybody in the audience, and don't forget that there will be another part of the school at 6 p.m.
Thank you.
Bye.
Goodbye.
Bye.
Thank you.
Bye. Bye, Rui. Ciao.
Ciao.